go through some of these clickers. So we're going to use this as a way to simulate discussion. So if something, you didn't get something and you want to discuss it, just raise your hand and let's discuss it. That's what these clickers are for. It's not assessment, it's for stimulating conversation and communication. Then, so we'll go through that, and then we will go through the next lecture. On Wednesday, I've pretty much decided, just to make the announcement, on Wednesday, we will start here, as always. I will send you to your breakout room, as before, and then we will come back here for a lecture after the breakout. So lecture, breakout, lecture, just like we've done before, to give us a break between lecture. But we'll come back, instead of doing a Zoom lecture, which I have found unsettling, we'll come back here. And if you would like to zoom in, you can zoom in. But if you would prefer to be in person, as many of you have expressed, we'll do it here. Okay? That's the plan for Wednesday. All right, what is mucus? Well, I'm just going to go through them and show you my answers. And you tell me if you have any questions. I'm going to try to do this quickly so that we can get to the next material. But again, I don't need to rush it if you have a question that we need to discuss and have a conversation about. And again, I'm going to pause, but I'm not going to go through. Raise your hand if you have a question. Zoom students, shout out if you have a question.
What about, like, why isn't D?
Why isn't what?
Why isn't D a correct answer?
D is, it's not really, that's a pathological, I mean, that's not its function. That's not why we evolved it. We evolved it to get rid of dirt and pathogens. And if you're infected, you produce more of it to get rid of dirt and pathogens, right? That's why you produce more mucus if you're sick.
Okay.
So I would not ask you a question like this on an exam. It's kind of vague. But the point is, these are the normal physiological functions. This is not a function of mucus. This is a function of the cilia. Was that your question?
Yeah.
Okay, here are the next set of questions. Let's just show the answers. Okay, I didn't teach you this. So this was, you could do this, well, maybe you couldn't do this without me teaching it. So let me teach it now. The cilia are an extension of the surface membrane. They are not similar to hair. They're not cellulose. So they're not all of the real growth. What they are is extensions of the surface membrane with microtubules inside. And the microtubules are what do the hula dance swift movement. What's the function of cilia? To transport mucus. Conducting airway. These three. Conducting airway. These two. I didn't teach really about where the surface area is great or where the volume is great, but the respiratory airway is where the surface area and the volume is great. Greatest. Conducting airway, goblet cells, ciliated, no, goblet cells and ciliated cells. Okay, let's talk for a moment here about the smooth muscle concept here. because I didn't teach that, and I should have, going backwards. Going back to this slide here. This airway smooth muscle is what controls the diameter of bronchioles. And that controls the flow of air into a terminal bronchiole, or into the alveolus. So smooth muscle in the airway controls the diameter of these tiny little airways, the bronchioles, the same way that smooth muscle controls the diameter of arterioles. So we have airway smooth muscle, and we have arterial smooth muscle, and venous smooth muscle. Airway, what we're talking about now is airway smooth muscle. Question? Yeah.
For when it says it is lined by cells that produce and move mucus, is that also applicable to the respiratory airway, since you had the respiratory bronchiole?
Is this the question we're looking at?
Yeah,
And which?
the bottom one, for C.
The diameter of some, but not all parts of it are controlled by smooth muscle, that's true.
For C, is it also applicable to the respiratory airway?
No, respiratory airway does not produce mucus.
Okay. Even though the respiratory bronchiole has cilia?
I'm not 100% sure that they do. The cartoon
Oh.
showed it, and that was what I'm not very sure that the respiratory bronchioles make mucus and have cilia that move it. Maybe they do, maybe they don't. That's not the key point. The key point is that the conducting airway does, and the respiratory airway is mostly involved in gas exchange. And let's, here, the diameter of the cell, of the alveoli is also not controlled by a smooth muscle. Notice, sorry, let's just scroll all the way back again. Notice that alveoli are not surrounded by smooth muscle. So these diameters are not controlled. These are. Okay, one more, I think. One more slide. What transport mechanisms move molecular oxygen? Okay, I told you that pretty clearly. Simple diffusion is the mechanism. Which of the statements about the respiratory airway are true? This is where gas exchange happens. And this is where we're going in the next hour. So this is the segue into, we're talking about surfactant now. You didn't know that until now, but now you do. Circle it, and we're going to talk about surfactant.
So,
Finish.
does that mean that the alveoli, since they're not surrounded by the smooth muscle, the surface area doesn't function for that?
Their diameter is set by development and how they grow, not by smooth muscle. Okay, so now I'm going to open up the other PowerPoint. So my intention is to load the new PowerPoint, the one with the answers, up onto Moodle. Right now the Moodle has the one without answers. I will endeavor to not forget, but if I forget to change out the PowerPoint, somebody please just remind me. Just all you have to do is say, did you remember to switch the PowerPoints? Obviously you know I didn't, and that will be my trigger to do so. All right, so like I said, we're going to go to surfactant, and now we're going to start talking about how the lungs move air in and out, the muscles of the respiratory system. So, in the old days, it was a couple of days between when we just talked and now. Now it's a couple of minutes. You remember this slide? You remember this slide? The type 1 cells are the cells that are specialized for this extremely thin layer for gas exchange. The type 2 alveolar cells are specialized for the secretion of surfactant. So these are the cells that secrete surfactant, and they secrete it into the lumen of the airway, and it lines. It's a thin film of molecular substance that we'll see in a minute that lines the inside of the airway. So these cells produce surfactant that lines the airway. That sort of lines the surface of the type 1 cells. Here is a cartoon that illustrates the airway. So here is the type 1 cell. Here's another type 1 cell. Thin, specialized for gas, for thinness, and for gas exchange. Let's not worry about macrophages. Here is a type 2 cell. Type 2 cells are specialized for the secretion of surfactant that lines the inside of the structure made by the type 1 cells. Now we're getting molecular. This is a molecular scaling. This is a phospholipid molecule. So surfactant, the head groups and the tails that you learned about in cell-phys. Surfactant is essentially phospholipid. It's phospholipid. It's a mixture of some specialized phospholipids. But it doesn't form a bilayer. It forms a monolayer with the hydrophobic tails facing air, which is relatively hydrophobic, dry, and the head groups both facing sort of a very, very thin film of fluid, water. So we have cell, water, head group, tail, and then actual gas. Why do we do this? Well, I'll tell you it's important. This is essential. Premature babies die because they are born before the type 2 cells produce surfactant. So extremely premature babies die from respiratory failure because these type 2 cells, it takes them, they don't develop until the end of the 3rd trimester. So if they're born too early, they die because they can't produce surfactant. That is the reason.
Do we know specifically which proteins now make it, so that, for example, we can do like gene editing or CRISPR, so that when they're born premature, you can actually
I
make
suppose
the babies make
in the
that.
future, we might be able to find either drugs that stimulate the existing type 2 cells, or maybe do a CRISPR and turn a type 1 cell into a type 2 cell. Yeah, maybe, someday. So the reason I give you that information is to try to illustrate and to emphasize the point that without surfactant, you would die. Why? It's physics. So this is weird physics, weird-ish physics. So I'm going to illustrate some physics. So what is this physics that I'm talking about? Well, what is this supposed to be illustrating? So here is a terminal bronchiole dividing into, here's a terminal bronchiole dividing into 2 respiratory bronchioles, and this is an alveolar sac, right? So this is a cartoon of the very, very end of an airway. The tube, the respiratory sac, okay? So this is a simplification of it. This is an air-filled tube, air-filled tube, surrounded by just a couple of cells. And the cells don't have a skeleton. They're flimsy as hell. They're flimsy little things. So there is the tendency to collapse, right? So now, let's think, let me illustrate physics. I keep saying it in a pejorative way. I love physics, but I know that not all of you do. Physics dictates that small bubbles have there's a thing called surface tension. You know what surface tension is. If you put a droplet of water on a hydrophobic surface, it beads up. And if you were to put a detergent into that drop of water, it would lower the surface tension and it would flow, right? So water has surface tension, and detergent or lipid or surfactant destroys surface tension, because lipid is like a detergent. So surfactant is like a detergent. And this is, we're not trying to clean the grease off of our pots and pans, we're trying to reduce surface tension. That's the mechanism of the surfactant. Why? Because of physics, two bubbles that are connected to each other that have different diameters, different radii, small bubbles have a higher internal pressure because of surface tension. And I forget the name of the French physicist who worked this out. I could look it up if you're interested. So, if we have two bubbles that are connected to each other, and one of them has higher pressure than the other, air will flow from high pressure to low pressure. And the point is, these bubbles are not, what we're talking about are alveoli. The alveoli are like bubbles in our chest. Air-filled films that are very flimsy, like a bubble. Small bubbles, small alveoli, connected to big alveoli, would dump all of their contents into the big alveoli. So if we have 10 smaller alveoli connected to one that's slightly bigger, the smaller ones are going to squeeze all their air into the big one. One big one has a lot less surface area than 10 smaller ones. You can work that out if you want mathematically. One alveolus, low surface area, respiratory inadequacy. Not enough surface area to do gas exchange. So now I want to do a little demo to prove that this physics actually, to make it make sense. So here is an airway and alveoli. Balloons are not perfect representations of bubbles, and so this doesn't always work, but let's try. So, and in the old days, I didn't have to wear a mask, so I'm going to try to do this safely by turning my back on you. Okay. Two alveoli. One smaller than the other. The prediction is, if this is a reasonable representation of the physics, this one is going to dump its, when I open this valve, this one is going to, I'm not going to blow into it anymore. This one is going to spontaneously contract, not contract, but the physics, the surface tension on this is going to create higher pressure in here than here. And air is going to move from here to here. That's the prediction. So it worked this time. I didn't blow this up with any higher pressure than this one. I blew them up with the same pressure. They're identical balloons. But because of surface tension, this balloon had higher pressure inside than this one, and it moved the air this way. And so now you can imagine thousands or thousands of smaller ones connected to one that's just slightly bigger And eventually, you get a situation like this. Why surfactant? Surfactant breaks surface tension. So the surface tension that created the higher pressure here is now broken by the surfactant. And small alveoli no longer have higher pressure than big alveoli because they no longer have lower surface tension. And you no longer dump air from small alveoli into big alveoli, and the alveoli stay inflated. So premature babies have an inadequate surface area of their alveoli because of this, because their type 2 cells do not produce the surfactant needed to disturb this surface tension dependent effect. Questions about that physics?
Would you say that if this physical property was not there, then we would need something like smooth muscle to contract these alveoli to dump the air into the other one?
I mean,
I'm just like thinking evolutionarily, like how, yeah.
yeah, but I mean, evolutionarily, physics comes before biology,
yeah. Okay.
right? We didn't evolve surface tension. Surface tension is. And we evolved in a world in which there is such a thing as surface tension.
Okay, so, I still have questions for this one, because even contraction would do a good job to do this, right? Like if we, but it would be,
Yeah, what would be the cost of, I mean, yeah, we could have smooth muscle that would prevent this from swelling. Just control the diameter. But now the smooth muscle is taking up area and reducing the area available for gas exchange. So that would be the cost of doing it that way.
makes sense. Okay, very cool.
So how exactly does oxygen go in and out?
Oh, we haven't gotten there yet. We haven't gotten there yet. We'll get there in a minute. First, we're going to talk about air getting in and out, and then we'll talk about how oxygen moves from air to blood. Okay, so now we're going to start on that. How do we get air in and out? So here, this is getting back to some anatomy. Here is a horizontal section through the lungs. So we're looking at a cross section. So let's get oriented. Here's the spinal cord. I mean, here's the backbone. So this is the posterior and this is anterior. This is probably the sternum right here. So sternum, rib, spine. Right? So this is someone, right one, so right, sternum, left. Okay, we talk about the root of the lung, also sometimes called the hilum, is where the airway, the bronchi, go into the lungs from the trachea. We're not seeing the trachea here, but we are seeing a bronchus here and here going into each lung. We saw in the lab, remember when we took the heart out? Well, some of you saw it, and some of you I told you about with a little bit flap of it. There was surrounding the heart, which is what used to be here, is the mediastinum. The mediastinum is that membrane that surrounded the heart and major vessels. The lungs are surrounded by two membranes that are called pleura. I guess one pleura. I've never talked about one at a time. The two membranes are called the two pleura, or pleural membranes. Plural, as in pleura. Pleura and pleura. So the lungs are surrounded by the pleura, and we didn't really see it in the cadaver. They're really fine, it's practically transparent. We might be able to see it if we look specifically for it. It's not small, it's surrounding the whole lungs, but it's very, very thin film, and we might mistake it just for extracellular matrix. It's surrounded by two membranes. The outer pleura is called the parietal pleura, and the inner pleura that's in contact with the lung itself is called the visceral pleura. So visceral pleura is in contact with the surface of the lung, and parietal pleura is in contact with the visceral surface on the inside, visceral pleura on the inside, and with the chest wall, the inside of the chest on the other side. So we have chest wall, parietal pleura, visceral pleura, lung. The pleura has different areas. There's the mediastinal area of the pleura, the diaphragmatic surface, because this is where the diaphragm is, and the costal surface. So these are all anatomical terms that I could test you on. Mediastinal, costal, diaphragmatic pleura. Also, mediastinal surface, costal surface, diaphragmatic surface. Costa, just because it's contacting the ribs. Diaphragmatic, because it's contacting the diaphragm. Mediastinal, because it's contacting the mediastinum. What's the purpose of this pleura? The pleura, visceral and parietal pleura, are filled with a very thin film of fluid, basically extracellular fluid. So, visceral pleura in contact with the lung, parietal pleura in contact with the chest wall. Very easy to move this way. Lots of lubrication that allow the chest and the lungs to move semi-independently in this direction. But when the chest wall expands, I can't pull these apart. The two surfaces, trust me, I would hand this around, but it's probably not safe. The two surfaces are connected by this vacuum, this partial vacuum, between the two surfaces, and the fluid fills that space. So I think I will break the glue that connects these knobs to the plastic before I would actually be able to pull the plastic apart, and I don't want to do that. So lubrication to allow lateral movement, but I can't pull them apart. So if this surface goes this way, the lung is going to go with it, because it's connected to the pleura, in the pleural space. But for example, so you may know that if you get an injury here, say a knife wound or some other injury here, and if you introduce air into this intrapleural space, I can now separate them, and now the lung is no longer connected to the chest wall by the pleura, right? So normally the lung is connected, physically connected, but highly lubricated to the chest wall. But if I introduce air, now the lungs can move independent of the chest wall. The chest wall can move and not inflate the lung. And that's a injury. That's pleural,
Effusion.
What's the word?
Pleural effusion.
Pleural effusion? Yeah. So that's the function of pleura. Now we have to get to Nahom's question. How do we get the air in and out? Well, you're probably aware, we use muscles. So let's talk about the muscles of normal, like what you're doing now, resting inspiration. The muscles required to just do resting inspiration are the diaphragm and the external intercostal muscles. Those are the muscles that allow the lung to inflate with air and inspire air. So the diaphragm is like a dome-shaped muscle, and when it contracts, it moves down like this. The dome moves down like this, and that increases the volume of the chest by pushing against the abdomen. So you may notice that if you're lying down and you're breathing normally and resting, your stomach goes up and down because your not because you're using your abdominal muscles, but because your diaphragm is pushing on your guts, inflating and inflating your lungs. So the diaphragm is the most important muscle for inspiration. By contracting, increases the volume of the chest, drawing air into the lungs. The external intercostals pull the ribs out a little bit, increasing the volume of the thorax, the chest, drawing air in. If you are exercising, you recruit additional muscle. You recruit the sternocleidomastoid muscle and the scalene muscles to lift up your clavicle and your first rib. And when you're exercising, you use additional muscles, so it's the diaphragm, the external intercostals, and the sternocleidomastoid and scalenes to maximally inflate your lung. So these are only recruited in the case of exercise. You're breathing hard. Those are the muscles that expand the chest, bringing air into the lungs. For resting expiration, you don't use muscles. You just relax. The lungs have a natural recoil. And so if they are not pulled open, they are not pulled outward, they collapse inward. And that is a resting expiration. It's just a passive process of the ribs sort of going back to where they belong, or where they were, the diaphragm relaxing, and the lungs' natural tendency to collapse.
What happens when you're like forcing breath out?
Okay, that's a forced expiration. So an active or forced expiration, what you use are your rectus abdominals. You push with your abdomen, and you use your internal intercostals. So when you are either, aware or unaware, if you are forcing air out, you're recruiting these muscles for a forced expiration. No?
So for this, when we're breathing out and letting air out, so is that the same thing as the two alveoli exchanging air? Like, is it because of the pressure difference?
No. So that's physics, and this is no, the force that moves air from one alveolus to another, if it's small and doesn't have surfactant, is an intrinsic property of surface tension and bubbles. This is a different physics. This is, by expanding this, increasing the volume lowers the pressure and draws air in. Contracting it increases the pressure and forces air out. And so that's actually in the next slide. When we inhale, contract the diaphragm, expand the chest, expand the volume. And so this is a cartoon of an alveolus, decreasing the pressure in the alveolus. If the alveolar pressure that is being decreased by expanding the volume around it is below atmospheric pressure, air moves in. Air moves down its pressure gradient. This is Ohm's law for air movement. So when the lungs expand, the pressure inside gets less. If it gets less than atmospheric, air moves in. When we exhale, the lungs' natural tendency to collapse and the chest wall natural tendency to collapse and the diaphragm's tendency to go back to where it came from squeezes the lung, increases the pressure in the alveoli, and when it's above atmospheric, it forces air out. So this is inspiration and this is expiration.
Bob,
Yeah,
can you go over expiration one more time?
expiration is, so under passive expiration, we don't actually use muscles to squeeze the lung, the chest, or to squeeze the lungs, but the lungs have a natural tendency. The ribs move back to their inspiration position. The diaphragm moves back to its to its inspiration position. And the lungs have a natural tendency to contract inward. Did that help?
Yeah.
So that's like, it's like we're squeezing the lungs, because we are, but it's not, we're not using muscles to squeeze the lungs. It's just the lungs have a natural tendency to squeeze themselves, and that puts higher pressure. You know, you squeeze something, you put pressure on the inside and forces air out. So here is a slide. This is kind of like the Wigner's diagram for respiratory system, but it's simpler. So we don't have to spend as much time with it. So let's just, what are we seeing here? So here's a cartoon of the lung, right? Costal surface, diaphragmatic surface, right? This is the parietal pleura. This is the visceral pleura. This is the intrapleural space, exaggerated a millionfold, right? It's only a film. It's not, it doesn't really have any volume, but it's there. So it's like a virtual volume. Visceral pleura, visceral pleura, intrapleural space, parietal pleura, lung, inside of the lung, diaphragmatic, costal surface, conducting airway, respiratory airway in this cartoon. That's this cartoon. This flinky thing is trying to illustrate the fact that the lungs have a natural tendency to collapse. Muscles are used to expand the chest, and when the muscles are not expanding, the chest is contracting. That's what that is supposed to be cartooning. Oh, I remember what I was, so I'll come back to that. So this is looking at air movement. So this is, so atmospheric pressure is, let's just say it's constant. When we inhale, the pressure in the alveoli is lower than the pressure in the atmosphere, and we get air flowing in. Air flowing in. So this is inspiration, and this is expiration. Expiration, the pressure gets higher inside the lungs and forces air out. Let's not worry about, well, no, I'm sorry, we have to worry about intrapleural pressure. So intrapleural pressure is always negative. There's always a partial vacuum because the lungs' natural tendency to recoil and retract. So if I just hold this one stationary, and this is visceral pleura on this side now, the lungs are connected to the visceral pleura. The lung has a natural tendency to recoil. When I pull on this, I'm making a partial vacuum in the intrapleural fluid between it, right? If I'm holding this one stationary and I'm pulling this way on this one, the pressure inside this space here is below atmospheric, which means it's negative. If I push on them, which never happens, then the pressure is higher than atmospheric in there. And if I pull them apart, it's a partial pressure, partial vacuum in this space. So there is always a negative intrapleural pressure. But during inspiration, I'm now pulling this way, and the lungs are naturally pulling this way. I'm making the inside more negative. And during expiration, the intrapleural pressure is becoming less negative. So the key thing is alveolar pressure is negative during inspiration and positive during expiration. Intrapleural pressure gets more negative during inspiration and less negative during expiration, but is always negative. Those are key physiological facts to know. And during volume, when we inhale, the volume goes up, bring air in, and then when we exhale, the volume goes down. Pulmonary physiologists like to talk about something called the transpulmonary pressure, which is a simple definition. It's the alveolar pressure minus the intrapleural pressure. So that's just defined. Well, and I might use that, but I tend to just say, well, enough pressures already. So let's start. So your book, let's do it the way your book says. Start at the end of expiration. So where are we? I'm going to expire. No, I'm going to exhale, but then I can't talk. Because I can't talk if I empty my lungs. So I'm just going to do it to show you. I'm going to exhale. Right there. That's where we are now. So at the end of expiration, I am no longer exhaling because the alveolar pressure and the atmospheric pressure are the same. Obviously, atmospheric pressure is not zero. We've got barometric pressure, but we call it, for this purposes, we say this is the baseline. This is zero. Atmospheric pressure, we're going to just call zero. Whatever atmospheric pressure it is, but we're going to call it zero now. That's our baseline. Alveolar pressure is zero. Intrapleural pressure is negative. And so transpulmonary pressure, zero minus a negative number, is positive. Okay, so now let me inhale. Now, I don't want to go all the way to the max. During inspiration, right there, middle of inspiration, muscles are pulling the parietal pleura outward, making the intrapleural pressure more negative. Alveolar pressure becomes negative. Air flows in. Transpulmonary pressure actually becomes more positive, but that's not the key thing. Yeah.
What would happen if you inhale or exhale above like what you normally exhale? So if I just taking a lot of air out of the ordinary.
Yeah, I'm maximally inhaling. That's over here. So that's I've inhaled maximum. Gone to you can only inflate your lungs so much. I can't go any further. I can't. I've stopped. That's the maximum I can go. Everybody's got a maximum.
Can you burst pop the alveoli?
No, I don't think so. I don't think we have the capability. I think you start to blow blood vessels before you actually burst the alveoli. I suppose if somebody put you out in outer space and the vacuum of outer space was sucking all the air out of your lungs, alveoli might collapse. I really don't know. So now we're going to where Nahum asked us to go to the maximal inspiration, end of inspiration. At the end of inspiration, no more air is moving. Alveolar pressure and atmospheric pressure are the same again. We've used muscles to pull the lungs to a maximum volume. intrapleural pressure is most negative. We've used our muscles to the maximum extent of that breath anyway, and intrapleural pressure is more negative. But there's no more flow because the pressure between the alveoli and the atmosphere is back to zero. So now we are here. Maximal negative intrapleural pressure, end of inspiration, no more flow. Now we exhale. The muscles are no longer pulling. The ribs are allowing collapse. The lungs tend to collapse. We generate positive alveolar pressure because air is flowing out. Positive pressure, air flows out. Well, not because. Positive pressure because the chest is pushing on the lungs, and because the lungs tend to collapse on their own. Positive alveolar pressure, negative intrapleural pressure, but less negative. Positive alveolar pressure, air flow, less negative intrapleural pressure. And then that's the cycle. Then we go back to where we were at the end of expiration. So that's the cycle of breathing in and breathing out. What do you need to know? Negative intrapleural pressure, negative alveolar pressure during inspiration, negative alveolar pressure during inspiration, positive alveolar pressure during expiration. Negative intrapleural pressure all the time, but more negative during inspiration and less negative during expiration. Those are the key things.
When you're at like a different altitude, would the system would be the same, just like the baseline is different?
Everything about this is the same at altitude. At altitude, we're going to talk, there's a problem set. I think it's on the problem set. Oh no, it's not on the problem set, but we will talk about altitude. At altitude, your respiratory system is being told to respire more because of low oxygen. And everything like this is happening, it's just happening more. Yes?
This is kind of going back to something you mentioned earlier, but with the, like you said, if there's a night balloon, they like air
Yeah.
in there, how do they, like, is there weights and then put the layers back together?
Yeah, there is. So what I was getting at there is if you break the pleural integrity, air can get into here, and that can cause a collapsed lung. Because now, this force, if air gets in there, the lung can move relative to the chest wall. And so air into this space can cause a collapsed lung, where this lung no longer gets inflated or deflated, it just collapsed. The way they do that is they can fix the wound, insert a little tube, recreate the vacuum, and by manually or mechanically making the negative intrapleural space, once they have sealed surgically or otherwise the wound, recreate the negative intrapleural space and reinflate the lung. So they seal the wound, create a negative intrapleural pressure mechanically, and reinflate the lung, and then things go back to, things work, goes back to fairly normal. Yeah.
What's the thing in movies where they like make a hole in someone's chest when they can't breathe, like put something sharp, like a pen or something?
trachea.
So are you talking about a tracheotomy?
Yeah. I think so, yeah.
If there's blockage of the airway, you can introduce an artificial hole in the trachea,
Oh, okay.
and then the person can breathe in and out, but instead of breathing in and out through their mouth or nose, they're breathing in and out through the tube.
Okay.
So I think I've told you everything that's on this slide. Any questions about this slide? So let's go to this slide, which is an important sort of in that, and it's both anatomy and physiology. What are we looking at? So the x-axis is time. This is time. Mark this on your slide. It doesn't, the book doesn't, nobody ever shows it, but this is time on the x-axis. And this is lung volume. So inspiration, expiration. Inspiration, expiration. Inspiration, expiration. Inspiration. Just someone breathe normally. The doctor said, I want you to go to a maximum exhale. Okay? So inspire and then breathe all the way out. Squeeze all there. Use everything you got. That's the maximum voluntary expiration. And then, okay, now breathe normally. Breathe normally. Now, at the end of a breath, at the end of an exhalation, I want you to breathe in all the way. It's hot. Inhale as far as you can. Okay? Inhale. I can't go any further. Okay, that's it. Maximum inspiration. Breathe normally. Go from a maximum inspiration to a maximum expiration. Right? Maximum volume to minimum volume. So these things have names. Names means it's anatomy. Let's parse out these names and we can fill in the box. Hint. Something that is a movement of change in the volume that is sort of unitary, is not comprised of two other volumes, is called a volume. Okay, so this is a volume, but something like this, that is comprised of this and this, is called a capacity. There's hint number one. This is a volume, it has a name, I'll give it to you in a minute, and this is a capacity. Is this a volume or a capacity?
Volume.
Volume. It's just this. So this is a volume, and where is it? If this is a volume, I'm looking for the one that goes from. So this is a capacity. Let's just use this one. Because this one is this volume plus this volume. Okay? This is inspiration, and this is expiration. Okay, so now let's attach some names to them. What is this volume that is just normal breathing? It's called tidal volume. No, don't follow me over here. This is tidal volume. This one right here. Like the tides. You know, sea comes in, sea goes out, the tides. Tidal volume. It's sometimes abbreviated TB, it's sometimes abbreviated BT. You have to, if you use abbreviations, you have to tell me what you're talking about. I usually use TB. Is this a volume or a capacity?
Volume.
Volume. Is
Wouldn't
it-
that be the most capacity that you have for inspiration? Oh, it's not a big hump.
We're going from the top of a normal inspiration to the top of a maximal inspiration. That's a capacity, because that's a volume, and it's got a name. Is it a is this volume associated with inspiration or with expiration? Inspiration. So it is an inspiratory reserved volume. IRV. Okay. Is this a volume or a capacity? Volume. Is it associated with inspiration or expiration? Expiration. So it is an expiratory. Just want to use a trick here. I want to make sure that this one isn't the trick. It's an expiratory reserve volume. ER. Is this a volume or a capacity? Volume. And it's called the residual. Volume. It's the volume that's left in your lungs after you blow out as much as you can. Even after a maximal expiration, there's still some volume left in my lungs. The residual volume. Okay, so now those are volumes. What is this? Is this a capacity or a volume? That's a capacity. Sure. Okay. Is it associated with inspiration or expiration? Inspiration. It is the inspiratory capacity. I see. Is this a volume or a capacity? That's a capacity. Is it associated with expiration or inspiration? Expiration. So what would you want to call it? If you, this is the trick. This is the trick. You would say, okay, if this is, it's an expiratory, following this, you'd say, okay, this is just the expiratory capacity. But it's not. They don't call it that. I don't know why they don't call it that. It's called the functional residual capacity. Why they did that to you, I don't know. Yeah, so they're
not just the capacity of
I guess, but you'd sort of, if you, yes, so now you explained it, because it includes the residual volume, so they included the word residual, but why not expiratory residual capacity in order to be somewhat linear with this? I can't explain it, that's the name. Why did they name it the heart? That's the name, it's the heart. So, functional residual capacity. Is this a volume or a capacity? Capacity. This is the capacity from maximal expiration to maximal inspiration, and it's called the vital capacity. That's an important measure of your lung function, because it's the volume your lung can move. It combines the anatomy of your lung with the anatomy of your